Okay, yeah, Acumen Pharmaceuticals is a clinical stage biopharmaceutical company, and we're developing sabirnetug, which is a next generation monoclonal antibody that targets amyloid beta oligomers. And so what we did is we completed our phase 1 study in the United States, and we did this in an Alzheimer's disease population. And what we found in our phase 1 is that the results were great. It led to the development of our phase 2 study. So we met our endpoints of safety and PK. And in designing the phase 2, we looked at that data to see that the number one reason for screen failures was amyloid negative PET scans. In the phase 2, what we wanted to do was to try and reduce the amyloid negative PET scans. And the way we decided to accomplish this was to introduce plasma p-tau217 biomarker and make it a part of the screening process. We decided on adding plasma p-tau217 at screening because as a standalone biomarker, it has consistently shown high performance in detecting Alzheimer's disease pathology in early Alzheimer's disease patients. And so our phase 2 study is looking at early Alzheimer's disease patients, and what we found is that by implementing this biomarker, we've been able to reduce the number of amyloid negativity, either through PET scans or CSF. And so what we found was that when we had all of the participants do the complete the p-tau217 plasma biomarker test, that about 50% came back as positive, 48% to be exact. What we did is we worked with LabCorp to develop a cutoff that we are calling an enrichment strategy in order to enrich the likelihood of getting amyloid positivity on either PET or CSF. And so that cutoff is 0.15, so that about 50% came back at the cut point or higher, which meant that they were able to move forward to actually, go to amyloid testing, amyloid confirmation testing. And so of those 48% to be exact who moved forward from the p-tau217 biomarker, it actually resulted in 78% of them being amyloid positive on PET or CSF. So we have taken, this has now resulted in about 7% of our total screen failure rate. And so when you compare that to our phase 1 study, where 60% of the screen fail rate was due to amyloid PET, we're reducing the burden. We're reducing the burden on the sites, we're reducing the burden on patients, and as well as us as the sponsor. And so, patients, about 50% of the patients who did not move forward then were spared the burden of often dreaded lumbar puncture or the unnecessary radiation exposure of a PET scan. So we've been really happy with the way that the results have turned out. We are still seeing amyloid negative screen failures, but we anticipated that. We expected that because, we wanted to enroll a heterogeneous population of early AD and not restrict it to a certain, maybe higher end p-tau score, which is why we, chose the cutoff at 0.15 that we did. So, next steps for us is, and one of the things I should mention is our phase 2 study is a global study. So it's taking place in the US, Canada, the UK, Spain, and Germany. But we were unable to use this assay in the EU and UK because it doesn't have a CE marking. And so, one of the limitations is, we're uncertain as to what the p-tau scores are at screening, and we're unable to use that as a screening tool to kind of, enhance or enrich the strategy or likelihood of getting that amyloid positive PET. And so that's, kind of a limitation. And so one of the things, of course, we'd like to study further is, kind of plasma biomarkers in general. There's a lot coming out in recent years about how, they're, being looked at as a possible diagnostic for using, a blood-based biomarker to diagnose Alzheimer's disease. So I don't think we're really ready for prime time yet in the clinic, but I think, after more research and more study, I think we'll be there before we know it.